Hello everyone. Today I'm honored to present you MM6 taxonomy that was recently published as part of the manuscript Fast and Sensitive Taxonomic Assignment to Metagenomic Contigs in Bioinformatics. As taxonomic analysis has many applications, I today want to focus on one. And this is its clinical use, wherein a patient sample is taken, sequenced, and then computationally analyzed. For example, to then identify a disease-causing agent, a pathogen. In this case, a tick-borne virus was identified in the patient's cerebrospinal fluid. The case report thankfully published the dataset, and we were able to build a tutorial out of that for our own method and generate this beautiful plot. To give you a high-level overview how our method works, first, after sequencing and potentially assembly, a lot of reads or contigs are generated. From these, we extract fragments, we translate those into proteins, and then do a homology search against the taxonomic protein reference database. From the best hit, we can compute a lowest common ancestor and then assign this lowest common ancestor to the read or contig. Okay, so what is MM6? MM6 is a homology search method with many search modes, BLAST-like ones, BLAST-P-like ones. It also contains capabilities for profile searches, similar to PsyBLAST or MR. It contains various translated search modes and also capabilities for nucleotide searches. It previously contained already a taxonomic assignment procedure for proteins against protein databases. However, in this work we expanded MM6 capabilities to make it more user-friendly by easy setting up, allowing it to easy set up and download and set up taxonomic databases. We accelerated the search and tailored it for taxonomic assignments for very large datasets. We added logic for taxonomic assignment to whole contigs, and various helper modules for visualization, for exporting of the results for downstream use. Okay, what makes MM6 special is its pre-filtering algorithm, through which the search space can be massively reduced, and very few true positives are lost at the same time. With only few sequence candidates remaining, more expensive algorithms such as Swift Waterman are not so expensive anymore. However, now in the taxonomic assignment we face a different issue. From one contig on both strands in 6 frames we extract a lot of fragments, and all of these fragments require homology search. To speed this procedure up, we implemented a fast rejection procedure, also based on the pre-filtering algorithm, however without its ungapped alignment stage to speed it further up. Here we take a fragment, translate it, and then do this pre-filter, and require at least 3 k-mer matches somewhere. If we don't find these, we reject the sequence, a fragment already, and further then we look at the diagonal where the hit was found and require an e-value of at least 100. If this also is not found, well, the fragment can be safely rejected, and the search is massively sped up, as only few fragments remain. Okay, so now having these fragments, we can search each of them with a slightly more sensitive search mode against the taxonomy reference database. For each fragment, we find the best hit in the database and retain the alignment region and note down the e-value to its best hits. And with this best hit aligned region, we can then repeat the search and find the candidates that have at most the e-value that we noted down in the first search. This procedure is called 2B-LCA, and it provides very robust LCAs. However, we have an issue here that we have to do 2 costly searches. We can save a lot of time here by not doing that, by doing only 1 search, as we already determined all candidates, all the most likely candidates that would be found in the second search anyway in the first one, and we have this pre-filtering results stored again. So instead of searching again in this second search, we can now only repeat the alignments against the candidates found previously in the aligned region, and go speeding up the method again. After having found all these candidates, we can then assign their weights, which are based on the e-value found, to their taxonomic label, and all taxonomic labels up the tree up to the root. We pick the most specific label that agrees to the majority parameter, which is by default 50%, and label then the contig according to its weighted vote. Here in this example, we found a bunch of Escherichia related sequences. What would be a good label for these? Well, family seems appropriate, as most of these are species. This one tiny fragment might just be a contamination or something. So we stick to a family label. However, as Escherichia coli and Shigella flexneri are actually very, very similar, here you see a fast ANI, a comparison of 2 genomes. Well, we could also give it a different label if we had used a different taxonomic database, such as GTTB. Escherichia flexneri might also be a valid label. And we also support alternative taxonomic databases, such as the GTTB. You're not restricted to only the NCBI taxonomy. Okay, to show that MM6 is actually as good as we claim, we benchmarked it on the Kami high complexity datasets from the first Kami challenge, and compared MM6 to CAT. It was also not long ago published. We evaluated using Amber, which is also part of the Kami challenge, in the taxonomic binning mode. You can see on the 2 reported measures from Amber, the completeness and purity measures, that MM6 and CAT are very similar in their performance. However, MM6 is massively faster. Another advantage that MM6 has over CAT is that it uses 6-frame translation instead of Prodigal. Prodigal, while it's very good, it's restricted to prokaryotic, to finding prokaryotic genes, which the 6-frame translation does not suffer from. Here we took 57 SAR RefSec assemblies, cut them into pieces so that they resemble real metagenomes, and then ran both with MM6 and CAT. And we see that MM6 assigns more context labels and more specific labels, and while being faster. To summarize, MM6 now offers facilities to easily download and set up taxonomic databases. It has a search mode that is particularly well suited to very large datasets. It has logic to assign robust taxonomic labels to contexts and scaffolds, and various modules to visualize and export results for downstream use. As you can see, with only 4 commands you can build and show this Corona visualization. I hope you will find MM6 useful for your research. Please look at the GitHub repository and the paper. I want to thank the whole team and also the amazing lab I'm in the studying lab, and all our funders.